We're still adjusting to the hybrid meetings, and they've been a little different each time. Okay, it's okay if it's, yeah, no, I think it's good enough there. All right, so here we go. All right, Guyana, because most people probably are wondering where it is, and I did too before I knew. So you can look at the smaller map up in the corner and see it's in the northeast of South America there, next to Suriname, Brazil, and Venezuela. So it's a very minorly populated country. There's only 786,000 people who live there. And it's not to be confused with French Guyana. That's another country bordering Suriname. And they were colonized by the French, and Guyana by the British and also the Dutch before that. And it is the only country in South America where English is the primarily spoken language. So there we go. So here's our great team that we went in December starting off and ended in January. We were there about a month. And I'll go into a little bit more detail about different members of our team and what they were researching. But just to kind of give you a quick overview, we have Dr. Kathy Aim in the front and center. And this whole research has been organized by her mostly. And then next to her is an undergraduate student at the University of Guyana, Alexis. Then next to her, we have one of our Amerindian guides, Luciano, and his brother, Sego, who's all the way up in the corner, and the sun kind of, can't really see his face there, but they've worked with Kathy for just about 20 years, going on these expeditions with her and assisting her out deep in the bush and setting up camps and collecting and doing all kinds of stuff. And then we have Delan all the way in the right. She's another Guyanese student, but she's actually getting her PhD at the Purdue University with Kathy in Indiana. And then Terry Torres Cruz standing up there on the upper right. She's a Costa Rican researcher, and she's getting her PhD at Penn State. And then we have Jack Johnson down squatting in the middle. He's my wonderful partner, fellow mycologist, and I can thank him for getting me on this trip. And then there's me. All right, so Dr. Kathy Aim is a renowned mycologist internationally. She's one of the coolest people I've ever met, and I'm really honored that I got to go on this trip and assist her team, be part of her team. And she's an expert on plant pathogens, especially rust fungi, and she does a lot of work looking at rusts that affect agricultural crops, and in the tropics, that would be largely coffee and chocolate. And she also studies macrofungi too in the tropics, and she surveys a lot of the fungi that are really highly underdocumented. A lot of tropical and rainforest fungi, there's such a diversity there, and it has been really poorly reflected in literature. And so that's something she's been doing for now just about 2 decades. And yeah, the fungi of the Guyana Shield and Gondwanan fungi are kind of part of these projects where they're looking at fungi of these regions. The Guyana Shields are really one of the world's oldest structures, like natural structures. It's like 1.7 billion years old, and it's this large like plate of earth, and it's like been degrading over a long time. And there's these huge waterfalls that kind of fall off of it. In Up, you know, the waterfall in Up, that movie, that's inspired by one of the waterfalls there. And the degradation of the shield actually creates really interesting landscapes where this red clay and this white sand that leads to a lot of interesting flora and fungi. I'm just getting used to my notes here real quick. Excuse me. All right, and yeah, so she's also worked with students at the University of Guyana. Every time she goes out, she typically brings a counterpart from the university there. And there aren't a lot of opportunities for the students there to study mycology. They don't have mycologists at the university. And so over time, she's developed really strong relationships with students and the natives who join her team as well. And here's a cool website you can check out. This kind of shows you more about the different ongoing projects. Terry Hinkle is another researcher who Kathy's worked with for a really long time, and they've kind of been working to map out the fungi of these regions. And the site is really fun to play with if you want to see some beautiful photos of tropical mushrooms besides the ones I'm going to show you, and kind of see some of those publications that have come out of this research. You can do that there. And then here are some of the new genera that Kathy and others have described. So, there's new species to fungi that, people find, and we're all, impressed, like, wow, it's new to science, but these are new genera. So this is even more broadly categorized in taxonomy. And these are all coming out of Guyana. And this is on Kathy's lab website if you want to see more about that. And there's, there's only 6 here, but there's definitely more than that, and dozens of species as well that she's been able to document. All right, I wish I could see this title thing. All right, so the Fusarium Xyris interactions. So this is Terry Cruz again. She's a PhD student. She's studying interactions between fungi, plants, soil, and insects. And she's specializing in this unique species of Fusarium that creates a pseudoflower on this species of Xyris, which is called a grass, even though it's not really a true grass. It grows in these grassy habitats. And Fusarium is really more widely known to be a crop pathogen that's destroyed like really important crops like bananas and tobacco and sweet potato, tomato, stuff like that. And that's, if you talk to a mycologist who knows about Fusarium, that's what they'll think of. They don't typically think of something like this, which on the right here, you can see that this fungus actually creates a fluffy pseudo flower mimicking the true flower on the left on the inflorescence of the Xyris. And so this is really a kind of a rare and unique thing for a Fusarium to do, and it hasn't been documented in any other species. And so Terry and Kathy and some others are trying to figure out, why does the fungus do this? It usually pseudoflowers. There are other fungi that make pseudoflowers, and it's typically to attract insects. That's why they would mimic a flower. And so we only recently learned that this fungus exists, and let alone what insects it draws in, and how the ecology plays out with the Xyris, because they're coexisting. How is it transmitted? Are insects carrying it, etc.? So that's what Terry's work has been, and I was able to help her in the field. I'll just show you a couple more photos. Oh, is this cut off? Oh, okay. Well, yeah, you can still see the flowers there. And then the pseudo flowers on the right on the bottom, and then they're kind of mixed in up there at the top. So you can tell the difference. The pseudo flower is much more of a bright orange, a bit more fluffy. But when you're out in the bright sun, and you're looking, it's like they look pretty dang similar. So it's really fascinating to witness. And here's just kind of what our research sites look like. They were open areas in the bright sun, a lot of flooded pools that kind of fill up when it rains and drain back out. But it's really pretty nutrient poor, white sand, and very open and grassy. And here is different things we did to collect data for Terry in the field. She's up there. The first 2 photos, well, top and bottom on the left, are collecting volatiles. So we're trying to look at what the flowers and what the fungus are emitting out into the air, and how, that tells us a lot about the physiology of the plant and, what it might be attracting. And then there's Jack along the transect. So we'd have to count tiny little spikes from these grasses along, I forget how many meters it was, but it was quite a ways. And we'd have to do it over and over again. So it was good mindfulness practice in the heat. And then there's just kind of like basically walking around. That's me just in the puddles, just constantly wading through all this water. But there was a lot of beautiful wildlife at these sites. You can see there's Drosera, so the sundew plants over in the upper corner, sphagnum moss in the middle, and reindeer lichen, these beautiful flowers in the Melastomataceae family. It's a new plant family to me, but they were all over the place. And then these tiny little frogs, they're little toads, little baby toads, and they were actually smaller than ants. That's a really close macro photo, but an ant walked by that and it was bigger than the frog. And I was really excited because I love frogs, and it's just incredible how small they were. So there was tadpoles in all the pools. And then that's like a fresh little froglet there. There was lots of dragonflies and bladderworts, that pretty yellow plant there. And then we were able to actually go into the interior. So the first sites I was showing you were towards Georgetown, which is along the coast. And then deeper kind of into the rainforest, it opens back up again and creates these natural savannas. And it's more of a natural habitat. The previous sites kind of had been more disturbed. And so it was really cool to see these really intact, kind of similar to prairie like that we have here. And so this is the Rupununi savanna, and it's in the middle of a village of the town Anai. Okay, so we spent most of our time in Guyana at the Iwakrama River Lodge and Research Center, which is pretty deep in the interior, surrounded by rainforest, but we were right along the river there, the Essequibo River, and it was this really cool structure we got to spend a lot of our days in. And I've got an arrow pointing to this window here because we spent many long nights at this table writing up species descriptions and processing specimens before they changed entirely, which even by the time we brought it from the tackle box in the field back and opened it up, they were already pretty different. So it's kind of like rushing against the clock to write up 20 species and measure their caps and take photos of them. And we were there into the night. We started our day at 6 a.m, go to bed at midnight after writing up a bunch of mushrooms. And through that window, tons of wildlife would just come right on in and say hello, bats flying over our head. There was lizards and giant moths and mosquitoes. And the whole table was just ants are just running around. And so it was really cool. You're really in it. And it's good mindfulness and a good challenge it's like mycology boot camp, really. I learned a lot doing that. There was a lot of river crossings and bushwhacking and a lot of mud. And so this is like actually specially modified vehicles that were basically all the vehicles that are in the interior have to be modified for this really muddy road, because they'll just fall apart within a week if you try to bring a regular car, because it's all dirt, like mud roads. It rains a lot, and then they're just super bumpy. And you need to have like special suspension on the car. So we'd pack 10 people in these little Land Rovers. They're actually pretty spacious. And then just a lot of the times there wouldn't even be a trail. We'd have to just we'd have guides who would help us like navigate. And we'd be looking on this big day where we were crossing a bunch of rivers, we were looking for more of the Xyris plant. And we hiked all day long, many, many miles the day before Christmas Eve. And it was not there, unfortunately, but it was a good test of endurance, and we saw lots of cool fungi along the way. Okay, Xylariaceae. So I'm going to give you a little bit more context about the trip, but a lot of the rest of the slides are going to be showing you different families of fungi that we observed the most. And we have Xylaria here, actually. We don't have as many species as there are in the tropics. And I got to see lots of really cool, like this yellow one and all these crazy spiky ones. But the reason it's first is because Delon Husbands, who is a Guyanese student, she's the one getting her PhD with Kathy in Indiana. She's studying Xylariaceae. And so we were particularly looking out for different species of these to collect and bring back to her for her to document. And in particular, the one that she's done the most work with is Xylaria caryoptera, and which is down at the bottom. And she actually described it with Kathy, and it's growing out of a big seed pod of green heart tree. And this is a really important lumber tree for Guyana. It's endemic there. It does grow like scattered kind of around, plants don't know country borders. So it does grow a little bit in Venezuela and Suriname, but it's mostly concentrated in Guyana, and it's really rot resistant. and lasts a really long time. And so it's highly sought after, and it's exported all over the world, especially to Britain, the former colonizer of the country. And so Delon was seeing that, so she was a forestry student, and they'd, cut down the green heart to process for timber, and then they're monitoring these plots to see if the green heart's growing back. That's the goal, have it be reforested. And they're finding this ilaria basically just destroying the seedlings, and not of all of them, but like a pretty massive amount of these populations were getting taken over by this fungus. And so her first work was describing it to science because it was not formally documented. And then now her more recent research is trying to figure out the life cycle. She was looking at like population genetics, trying to see, is it one species? Is it multiple? How many populations are there, et cetera? So it's a pretty crazy looking fungus, and I think Xylaria are awesome. We have the most common species here is the carbon antler. So you might see like kind of similarly branched looking mushrooms in our forest as well, but they're not on seeds, they're on wood. And then here's Luciano and his Xylaria collections. And this was in like 30 minutes. So Luciano is, born and raised. He's a Patamonia Amerindian. He spent his whole life in the interior rainforest. And he is so skilled and also intuitive at just working through the forest. And if you say, Luciano, can you look out for X, Y, or Z? You could show him anything, and he's within, an hour, you've got more than enough specimens. So this is him collecting Xylaria for Delon, and they've worked together for quite a few years. So it's cool to see that. So familiar fungal forms. So here's some mushrooms that I saw that I was like, I recognize what that is, and it looks like what it is in my mind. And I put that first because a lot of the mushrooms I was seeing down there, I'm saying, Kathy, what is this mushroom? And she's saying, oh, that's, let me see, what do I have here? That's a gymnopus. And I'm like, that doesn't look anything, it's supposed to have gills and X, Y, and Z. And so these are the mushrooms that I saw that look the way that I recognize them to be here or in literature or whatnot. We have an amanita, jelly tooth. We were just talking about jelly teeth. There's a bird's nest down there, Cyathus, and auricularia, Callistosporium, and Schizophyllum. And then there's just a lot of new and unusual fungi to me too. So, and I knew what I knew that this was going to happen. I had been looking on that tropical fungi site, looking at some papers and just having my mind blown every day because, you don't expect a lactarius to grow, looking like an oyster mushroom, but that's the way it is down there. Things are different, and it's cool to see the overlap and how things are different. And so there's my first stinkhorn I got to see. They do grow in North America, but I had not ever found one. So I was happy to find that. And up in the corner is a mucronella. So the mucronella we have here look like little tiny icicles that kind of dangle, the kind of spine fungi. And this one they call it yellow snot because it looks like a blob. And then here's one of the few ectomycorrhizal fungi we saw in the middle, this jelly meatball. It's called Tremelogaster surinamensis, and it basically looks like meatball that if you cut it open, there's caviar and then purple powder in the center. And it's associated with, I think, the coccoloba tree. And this amanita too, going back for a second, is also ectomycorrhizal. And the reason I'm pointing that out is because most of the fungi we saw were not mycorrhizal because of the forest types we were in and the elevation we were at and the seasonality. So we only documented a couple of those, but the jelly meatballs were really cool. And then the bottom left, so that's what I was saying. That's technically a gymnopus, and our gymnopus that we have, they get shuffled around all the time, but they have gills, and that's what you would think of, not this like weird cone. They call them pod parachutes. So it was kind of like one of my first real, like, tangible ways of holding something and be like, this is so unusual. And then Delicatula, these tiny little mushrooms in the Trichiloma family that have no gills. They're kind of tiny little crystalline They have a cap, they have a stipe, but there's no gills. And then I'm not going to give you each species each time, or this will take forever, but I want to tell you about all of them. All right. How's everybody doing? Talking good? Okay, there you go. So small saprobic, that's the categories I would put most of the mushrooms we found in. Most of the mushrooms we found were like less than 5 inches long. And we love, Jack and I personally love tiny mushrooms. So it was great. And Kathy was impressed that we were, so consistently excited to be finding them, because a lot of people like bigger charismatic mushrooms, and the tropics do have those as well. So it worked out, but most of our time was spent measuring like centimeters and millimeters of these tiny mushrooms. They were also mostly saprobes, so just decaying the leaf litter, twigs, branches, seed pods, leaves, any vegetation was, mushrooms we found were typically decaying them. There's a cool seed pod down there in Trinidad that is covered with Dendrostabella, or a similar fungus, which is like this crazy alien looking thing. Okay, here's Trinidad and Tobago. So after the month in Guyana, Jack and I left the team and flew over to meet Luca in Trinidad, who Luca has family there. Their mom grew up there. And so we had a really cool trip, naturalizing, meeting other scientists, and kind of like mimicking what we had done in Guyana more formally, and trying to practice documenting fungi in the same nature, on our own accord. And it was a really cool to go from one to the other. And so Trinidad is actually right off the coast of Venezuela, just 7 miles off the coast. It actually pretty recently separated in geologic time off of the continent. And the country is made up of Trinidad, the big island, and then Tobago, the further up island, which unfortunately I didn't get to go to, but apparently everything that grows there is giant compared to the ones on Trinidad. And then there's a chain of smaller islands in the middle between Venezuela and Trinidad called the Lesser Antilles. And something that's really cool about this island, and that makes it a little bit unique, is you can see in the map, it says the Northern Range. So that's a mountain range, and it's actually the furthest tip of the Andes. And most people don't, even people on the island we were talking to didn't realize that. And so the habitat there is more similar to the inland mountain habitat in South America. And it varies as you go through different parts of the island. And we started off kind of going right into it. We made our way up to just about the highest point of the highest mountain in Trinidad. I don't know the name of it, but we stayed at this incredible place called the Cocoa Palace in Brazo Seco village. And we just had a great time getting out right away, documenting things. We met a bunch of researchers right off the bat who we had known from online and were actually already planning on meeting up with. And by chance, they were leaving as we were coming, and this after driving through the mountains for hours. And they're herpetologists, so they study reptiles, but they also study insects, and they were setting up camera traps to document ocelots. So those interesting wild cats. And yeah, there's lots of waterfalls, really cool lianas, these woody vines, and creeks and whatnot. And we had a good time. I'm just gonna find where I am in my notes. Thank you all here. Okay. So yeah, it's a really lush place. It's absolutely incredible, really affordable to stay there, and lots of researchers go there. So if you're looking to do that, check it out. We collected and deposited 134 specimens to the National Herbarium of Trinidad and Tobago, which is at the University of the West Indies, St. Augustine campus. And they had not had specimens of fungi deposited formally since the 40s. So this is I didn't take the pictures on the left, but on the right, you can see this is a bird's nest collection from 1947. And all of these folders, all of these cabinets in the photo with these women in it, each are dedicated to their own plant family. And so, and there's rows and rows of them, and that's standard for an herbarium. But all the way in the bottom corner, there was a shoebox. And there was a couple of fungi collections that were not actually formally deposited in there. And then there was one folder that said lichens, and then this bird's nest was in there. So clearly it has not been given attention for a long time. And that's not entirely the fault of the institution, actually. Well, there was a curator of the herbarium who, when people asked if they could work with fungi, she declined them. So that really stunk to hear people who had been excited about mushrooms and been turned away. So we were trying to help get that changed, and we got to work with some botanists at the college and deposit specimens. And it was just a great process and really cool to see how these different colleges handle their specimens. And also just in terms of giving you the stats, we had, so me, Luca, and Jack had 134 specimens in Trinidad. I had the least. I was kind of like, after Guyana, I was like, I'm taking a little bit of break here. Still did a handful, but I got to give them more credit there. And then there was Jack and I together had 216 in Guyana that we deposited, half at the university, and were able to have go half back to Purdue. So those are mushrooms we photographed, documented, dried, and preserved. So here's Jeffrey Wongsong. He is Trinidad and Tobago's mushroom man. And if you talk to anybody about mushrooms on the entire island, they will tell you, do you know Jeffrey? And we had already been talking to Jeffrey because the people we knew on the island, Luca's mother and family, had already like gone to school with him in the past, and everybody knows each other. And he was super excited to meet with us. He doesn't meet a lot of mycologists or people who want to really seriously pursue fungi as much as he does. And so he actually has a mushroom museum that he runs out of his house. And he has hundreds of preserved mushroom specimens in jars like this. And so look how well they're preserved there. That's the mushroom that's been in there for a long time. And it was really impressive to see. None of these, unfortunately, are going to be useful for doing sequencing work. So we were talking to him, kind of finding a middle ground there, because he loves the aesthetic of the jars. But he was a great man, and he runs a Facebook page that you can check out, Mushrooms of Trinidad and Tobago. And people share their photos and they talk about ID, and it's kind of the only place they have to do that. So I just have a lot of respect for him cultivating community there. All right, taking a breather. Woo. So the botanists that we linked up with at the herbarium and the university helped us get the permitting to be able to visit the protected Aripo savanna. So similar habitat to the savannas in Guyana. There's less of them preserved in Trinidad than were native there. And so you have to have permitting to go, and it was really cool to see a lot of the same plants that we had seen. We spent most of the time actually in the forest surrounding the savanna because there was so many waxy caps, more than we had seen really at once during the entire trip, including this incredible green one that I had been told to look out for by the Jean Lodge, Dr. Jean Lodge, the waxy cap expert. And she studies tropical wax caps, and I was hoping I'd see a bluishy green one. Most of the ones I saw were red. So this was exciting to find. And I'll show you some more of those. Here's the mangrove, the Caroni swamp. It's the second largest mangrove in Trinidad, and it is a bird sanctuary that has, it's protected because of the scarlet ibis, and they were poached and then protected to preserve the remaining populations. And they're just beautiful, bright pink birds. There's flamingos, there's boas in the trees, there's crabs crawling up the vines. It's a really spectacular place, and you have to go through by boat, and the tour guides actually are actively involved in conservation and nature education, and it was really cool to see that. All right, I'm going to show you mushrooms now for the rest of the time. Here's the waxy cap family. Everybody knows by now I love the waxy caps, as do many in our club. Oh, got some feedback. Someone unmuted. Okay, so like I said, most of them are red. They were uniquely red to me. Waxy caps, you start to notice, different shades and hues and gradients of red. It gets hard to describe it in words after the 100th time, but it was really nice to see them in the Xyris sites, because when we were out in the bright sun, there really weren't many mushrooms in the sandy plains, and that's because it's very nutrient poor. And waxy caps are okay with that. So there was a high tendency of wax caps, and we got to see at least one a day at the few days along the Xyris, but tons in the interior. Tiny and Alone, A Tale of Tropical Hygroscopies. And so this is the reality of, I'm all gung-ho and I'm like, I'm gonna go study wax caps down in the tropics, and there was so many of them. There was more than I could collect. And it's also kind of, sometimes I wonder, like, why, wax caps are so beautiful and amazing. Why aren't, more people studying them? And, it's this, I was shown that kind of going down here, because a lot of them are just a single mushroom. Kathy called them singletons. And she's like, try to avoid the singletons, because having multiple specimens is really helpful and pretty important for microscopy and studying the macroscopic features for sequencing, because these are single mushrooms. Look at it in my hand, and that's fresh, and then it dries down to just barely anything. And so to be able to have enough gills to see spores and measure the spores is tricky. And so I have to try to keep track, like this is an example, red hygro 1, red hygro 2, and we're supposed to be coming up with field names to document them, to keep them separate. And it was really challenging after a while. And all of them are undescribed. So there was no names to call them. It was fun, but it was hard. And so I just thought I'd kind of show you what that was like. And this was kind of more of a rare occurrence, when there was enough mushrooms for me to, one, lay them down and take a picture together. And then more importantly, to have multiple tissue samples to work with. And these were some of my favorite finds. The one on the right here is really cool. I called them sand funnels. And after miles and miles of hiking in the bush, came out, and there was just like 30 of these few centimeter long little red waxies, and they kind of have these veiny gills and the super flat top. You can balance them right on their head. And that was probably my favorite find of the whole trip. And they're even kind of like club shaped there. So, and we know that there are waxy caps that don't have gills in the same genus Hygroscopy. There's one from Ecuador. And so it's kind of like maybe an intermediate, veiny gills. So I got excited. And then the Hygroscopy conica group, these are like the witches caps that are also in North America. We have our own species Singeri here. We know based off of the South Sound studies that there's at least more than one. I don't know if there's more than two, but we think so. And they're really hairy. So you can see how much, it's hard to see it on the screen here in person, but there's all the caps of them are pretty variable. They stain black just like they do here. And it was just fascinating to see how hairy they could be for these. And they were pretty small. So that's all I'm going to say about those guys. All right. And Entolomataceae, pink gill family. We have lots of entelomas here as well. But basically Entolomataceae and Hygropheraceae were the most abundant, I'd say, at a lot of our sites, which is exciting because they're two of my favorite families. And you can see the gills kind of turning pink on some of them. They have pinkish buff spores. And there's a genus Inocephalus that is in Eastern North America and also down the tropics. And they have these really funny pointy caps. And it's just it just looks like somebody stuck a little horn on these mushrooms. And they're called unicorn entelomas in the East. And I got to see one blue one. I was hoping I'd see a blue one. And I kind of was walking all day. And I was like, I think I'll see a blue enteloma today. And like, miles later, I'm walking and we're not on a trail. And I look down and there it was right in front of my feet. And I was like, I got my one blue enteloma in. Okay, Mycinaceae. So another family that's really abundant here as well. But there's tons of them in the tropics, leaf littered decomposers all around. They love the leaves. and the palm stems and wood, and they look pretty variable. They're variable here as well, but like this one up top, this favolacia, these tiny little honeycomb-like mushrooms, you'd never think are in the same family as these other guys, but they are, and they're super small. I think like a few millimeters, or maybe 1 millimeter for those guys. Really small and spectacular. And these red ones were also really common on the leaf litter. Marasmiaceae, also just this probably I think was the most abundant of all the mushrooms we saw. This family, there was just everywhere you looked, you'd find some of these. They're called pinwheels or parachutes. And I think they are a lot prettier down there than they are up here. And they had some forms that I was not familiar with. This cool one on the bottom, Cadiocalathus, kind of grows as a pleurotoid growth form. And up in the upper left, this is Bruniocorticium, and doesn't look like the other ones at all. And it's because it's actually not making mushrooms. It lives entirely in its asexual stage. And it makes these nodes along like the center parts of the leaves. It actually creates like a mycelial web up in the canopy and will catch leaf litter, eat them down to the bare strip, the harder strip in the center of the leaf, and then make these little tiny nodes that get kind of plopped off and spread that way. And birds actually use them in their nests, and because they'll make long strings of them the way they grow, and the birds can weave them into their nests and potentially create an antimicrobial situation there. But the pink ones were really beautiful. All right, cordyceps. Entomopathogens, everybody loves these. I guess warning you now, if you haven't already been warned, the next couple of slides have a lot of insects on them and a lot of insects being eaten by fungus. So if you're not a fan of insects or fungus or both together, you're not going to want to look at this. These were definitely the most impressive cordyceps I'd ever seen. The one in the middle was most exciting to me. This is Cordyceps tenuipes, and it's actually growing on a leaf cocoon surrounding a larva that was underneath the duff a bit. And so all I saw was this yellow frilly parts coming out. It's like brown leaves. And then I see the structure, and I saw it and my brain was like mushroom, but then I have to tell yourself, be prepared, because it's probably a flower. There's lots of flowers in the rainforest, and there's lots of plant matter on the ground. So you have to just be like, it's very likely also just a piece of a plant. And you've never seen most of these plants. They have all different crazy parts. And I was really excited. I was like, I'm going to dig it up, because that's what you'd want to do. If you find one of these, you want to be able to see what insect it's growing on. And luckily I was right. And I got to preserve a couple of these actually. And then Luciano found this beautiful Ophiocordyceps amazonica, the pink species growing on a cricket, I forget. And then I found this one growing on an ant as well on the left. It's in an asexual phase. And so is the one in the middle. The one in the upper right is the only one that has is in its sexual stage. And then on the bottom, there's more than anything, the most common entomopathogen were these like spots on the underside of leaves. And you'll see that here as well, where the fungus kind of plasters the insect that it's eating to a leaf. And hypocrella were what these are called. And we shined a UV light on it just to kind of see what it looked like there. And just common orange spots on leaves. But they were everywhere. Here's some crazy ones. This is from Trinidad. Those other ones were from Guyana. This is a night we spent out with the herpetology group, and we got to see tons of reptiles. I held a bunch of snakes, held a tarantula. They're actually really cute. They're tiny ones with a heart on their butt. And we saw they spend a lot of time looking at insects and live moths and such. And so they're super experienced. They're like, oh yeah, you guys want to see some cordyceps? And they just walk right up and they're showing us on the leaves some of the coolest cordyceps like we had ever seen. So it was all within like an hour, we saw these. And the spider on the bottom right is very large. It's like that big. It's a huntsman spider. And there was a couple of these that were being parasitized by Terubiella fungi. And the one all the way in the bottom right actually has a hyperparasite on it. So there's the fungus growing on the insect, and then there's another fungus growing on the cordyceps's fruiting structure. So that was something that kind of blew our minds. And then we shined a UV light on the Terubiella, and it looks like it's from a horror movie. It's pretty cool. You can see the fungus behind the eyes. And then here's some cool insects that haven't been eaten by a fungus yet. You know, they probably will at some point in some way or another, but just to kind of make up for all the gore. And I, after this trip, appreciate insects a lot more than I ever thought I would. And I got to see a lot of beautiful butterflies and moths and spiders and pretty hardcore looking orb weaver in the corner there. They were just really spectacular to witness, especially at night. I like night walks. Okay, leafcutter ants. All right, so let me see if this works here. So, common sight when you're walking on in the rainforest there, you see these little guys with these huge leaves kind of crawling, and you're like, where are they going? You know, what are they doing? There's so many of them, and they're clearly on a mission. And I knew about leafcutter ants, but I didn't really know how cool they were. They actually have really complex societies, and they're bringing that plant matter. They can carry up to 20 times their weight, and they're bringing it back to their colony to feed a fungus that's going to feed their babies and the adults as well. But they're making baby food with that from a fungus. And here's what some of their mounds look like here, the bottom left. And you can see, actually, it's a little bit hard to see on the sand, but these little guys clamped onto this leaf matter. And they're pretty non-specific about the leaves they bring back, but they take signs from the fungus. So if they bring a plant that the fungus is toxic to the fungus, they can detect the compounds that the fungus is putting out. That's like a stress hormone, and they will not ever bring it back again. So they're like super in tune with their system. And I actually have a slide here showing you a little bit more about their symbiosis, because I'm just amazed by it. So the farming ant will bring back the leaf to feed the fungus that's kind of living within the underground structures that they live in. And they're actually co-evolved over 30 million years to make specific structures that the ants can then plop off and more easily eat and feed to their developing larvae. This is so cool. I'll give you one more fact at the end of this. But so the ants carry on their bodies a bacteria that is actually producing an antimicrobial compound that's fending off this crop pest. So another fungus that's actually a parasite on the fungus that the ants want. So it's this mycoparasite, this fungus trying to eat the ants' fungus, and the ants are carrying around the the antibiotics basically to kill off this other fungus to keep their food source safe. And it's just, I think, one of the coolest things ever. Sorry, I'm just like in awe. I was just reading more about it before doing this presentation, and it gets cooler and cooler. And one more thing is that I didn't know is that the fungi that there's many species of fungi that these ants interact with, but they're all in Agaricacea. And none of them make mushrooms anymore. So agaricus is like Agaricus bisporus, the mushroom, the button mushroom, Comini, portobello, that's an agaricus. That family is getting moved around a lot, but essentially fungi that are related to those that, don't need to make their own mushroom, but instead make these little structures, Gonglydia, I think, for their ants to feed on. And is there another thing about them? No, just long terms of time of coevolution together. And there's 47 species of leafcutter ants. Here's just a little bit more on the interactions of fungi and insects. Last slide, I think, on that topic. You can see different termite and ant mounds that fungi are helping just to decay. I found this really pretty, I think, Clitopilus growing on, I think it's a termite mound in the upper left, and kind of zoomed into the mushroom there. And then other mushrooms growing on these clumps that were like an abandoned ant's nest of some sort. And then you can see lichen, powdery lichen growing on also a former mound. All right, Ascomycetes, cup fungi. There was not a crazy amount, but the most common ones we saw were these really cool pink species down at the bottom, Cucina. And I have wanted to see these for as long as I thought mushrooms were cool. So like, I don't know, 10 years. And they're really, I didn't know that they were just so abundant there. And there's actually like, you can search photos up and see like tiny frogs that will like, there'll be water that will fill up in these cups, and the tiny like poison dart frogs will kind of chill in them. We didn't see that, but lots of coquina, common orange cup fungi, weird earth tongue like mushrooms in Trinidad there, and also this cool orange. I'm using cool a lot. This is mostly being like, wow, aren't all these things cool? More than trying to like, do ID dive in like I normally do. But they call it orange bladders, and this I cut it open, but it's normally one piece, and it's this wrinkly, pretty big ball, and it's this strange asco that's pretty common in Trinidad. And then you can see the one in the center, it's the tip of a leaf petiole. So there's certain ones that will just grow one fruit right at the end of a leaf. I thought that was kind of a sweet ecology. Polyparales, Regina, this one goes out to you. Lots of polypores, and I have it as the order here because there are many families in Polyparales, and so these are mostly pored mushrooms, but they're not all pored. In the center, we have a gilled mushroom, actually, Lentinus, I believe cranitis, or this is definitely a Lentinus, and they are edible. Some tribes will eat these, and just lots of polypores, some of them that looked kind of like polypores we have here. You could see the one, that kind of looks reminiscent of a Ganoderma that we have, but I don't even know if it was a Ganoderma, but kind of looks like it is. And it was growing on these huge, huge buttressed mora trees. And those were some of the biggest polypores that we got to see. And then tons of these bright orange Trametes, and a lot of stalked polypores. You could see it's kind of hard to tell, but look at this raised platter of a polypore in the upper right corner. The honeycomb favolus, which I didn't know were in Polyparales, and they're like pretty fleshy mushrooms, and they have the tiny ones in Mycinaceae I showed you, the Favolacea. I thought they were related, but not very closely. And then these wiry looking fruits down at the bottom are actually Polyporus guyanensis, and they're everywhere. And they're these long, like sometimes for feet, stringy, upright structures, and they're asexual growths of this polypore that will only very sometimes make a tiny little pored sexual fruit. And it looks like it's a xylaria or some kind of like rhizomorph of a marasmius maybe, but they are polypores and you would really never suspect it. And they're very tough and wiry. Clubs and corals, we didn't see too many of these. The photo in the middle is a friend, one of our herpetologist friend, Kaima's, but it was one that I have saved that I hope to see in the future. It's kind of a bluish gray coral. And then, some other ones there, these orange ones are really cool, but I will not divulge too much because they need some further study. But I just thought I'd show you guys a picture anyways, because they look like standard corals. And, maybe they are, maybe they're not. Crust fungi. The one in the upper right corner, the word crust fungi, everyone has their own definition. I've recently had mine double checked, and it's basically just something that like a fungus that the fruiting structure is smooth. But I thought I would include this tricaptum like fungus. I think it is a tricaptum, just because it reminds me of crusts, but this cool blue one, I'd never seen a blue crust before. And a lot of this yellow mustard crust, they were calling it, was on a lot of the wood there in pretty big swaths. And I like crusts, so Right, we're getting to the end here. Lichens, I really like lichens. If any of you were at the talk, was that last month? Feels so long ago. Last month's talk by Dr. Fred Rhodes. He gave us a lichen talk. And yeah, I've also studied lichens and didn't document them to the same rigorous extent as the mushrooms, but definitely observed them and was fascinated by all their different substrates. You can see the palm leaves. There's moss and lichen just growing on these palms. A lot of lichen on the tree bark. There was this black kind of the light shining through. These are cyanobacterial, kind of more gelatinous lichens that were really abundant in Trinidad because there's a lot of liquid water there, and cyanolichens like liquid water. And this beautiful script lichen in the middle at one of our Zyra sites. And then just more is a basidio lichen with clubs down at the bottom. We actually have one here as well. Is it Mucronella? No, multiclavula. Sorry if my breathing is kind of intense there. And then some cups on a jelly lichen. And then something that I didn't know was a lichen all the way in the upper right corner, Cynogonium. I wasn't sure if it was a bryophyte or algae, and it's algae and fungus together, making these little green brackets all over the place. And then some slime molds. Most of the slime molds we saw were Serratia mixta species. We have this one, if it's the same, Fruticulosa on the left, the larger photo here as well. And then I got to see some new ones that have this clear, liquidy stalk. And they were really tiny and fun. Lots of plants. I could have made a whole slideshow about the plants. But here's just some highlights. There's a mycoheterotrophic plant in the corner, the yellow flowery thing, very tiny plant. There's epiphytic cacti just everywhere, Ripsalis in the middle. They're just dangling off of the trees in large abundance, like in urban areas. So that was really cool. That's a fern in the upper right corner. Those are all the sori where all the spores are made in this beautiful pattern. A flower, I think, is aptly called a clitoria. And then there is a flower emerging off of directly from the bark. I don't actually know how this flower works. I just learned what it is, but I don't know how it does this. It's just very cool. It just pops right off of the tree there. And then that's a cashew. That's my first cashew I ever saw. And that's a big fruit. And then you can see that like inside that pod is the nut, the cashew nut. And people make juice and eat the fruit, the fruit. And it was actually in flower too. You could see the little whitish pink flowers. And that was really cool to see a cashew. And then lots of animals, more insects, because they're great. Frogs, they're frogs mating, if you can't tell. This is called amplexus, when they hold each other for a very long time. I don't actually know how long, but they hop around together for a while, and it's actually the male on the top. And these are cane toads, and they're everywhere in the tropics. They're invasive in Australia and other places, and they wreak havoc there, but it's nice to see them in their native range. They're called crapaud, which is French, I think. And they just come into all the indoor rooms. So like you'll be working and then you'll look over and there's like one almost all the time looking at you. And they're kind of doofy. And then black caiman. So I actually fed that caiman chicken on Christmas. The natives who run the lodge do it regularly, so I didn't like step in and say I'm going to feed this thing. But it's that one's name is Sankar, and he comes every day, or she's missing a leg, and they help her out, and everybody loves to see her. And it was just incredible to see a giant caiman. It was just unbelievable. There's a really cute snake, tropical flat snake, these crazy insects. I'm forgetting what they're called right now with the fluffy ends, but that was quite a sight. Jaguar footprints and a weird marsupial that freaked me out. And we freaked it out too in a tree there. All right, resources. We're just about done here. Here's some things you could look further into it. Definitely, if you're really trying to dive into tropical mycology, look up research by Kathy Aim. Her full name is Mary Catherine Aim. Terry Hinkle, Deborah Jean Lodge on the Waxies. She's done other tropical work too. Biddy Roy, who's based in Oregon. Noah Siegel, who I actually got to meet up with. I think he's attending this talk right now. Hi, Noah, if you are. But we got to meet up in Guyana, and that was just really cool. He was actually on Terry Henkel's team. So Kathy and Terry have gone down there for a long time together and now do separate endeavors. And our two guides, Luciano and Sego, went from our team to their team, and they did a full bush camp. We stayed at a lodge, so they did a little bit more hardcore, pretty much a lot more hardcore. Anyways, Danny Newman and Rue Vandegrift also have done good work in the tropics. And tropicalfungi.org. There's Kathy's website, the Irokrama Lodge website that has some cool resources on birds and plants, little guides you could print out if you wanted. And then there's the Mushrooms of Trinidad and Tobago Facebook page. Thank you very much for being here. There's my email and my INAT and my Instagram. Thank you guys. I actually have some quick trivia. Do we start still giving prizes away for that? All right, so you can participate in this if you want. There's just 3 questions and you have to be here live. And you'll win a prize if you get it right. Okay, so this is very generally, who are the 4 participants in the leaf cutter symbiosis? Does anybody know? Just like main players. Raise your hand. Yeah, you said the fungi, the bacteria, the ants and the crop pest, which is all what? What is the pest? Yep. Woohoo, Gina. Am I giving this?
For everyone at home, she wins brain energy from Host Defense. It was donated to us from Fungi Perfecti.
Woohoo. Great. Thanks, Regina. There we go. Ants, bacteria, and 2 fungi. And there's more involved, really. They're spectacular. They're just right. And you have to it's like the Wikipedia is like, these ants have the 2nd most complex societies after humans. So like they're in their whole highway. And I'm like, whoops, like trying to avoid stepping on them because they're so impressive. Great job on that. We got 2 more. All right. What structure does the fungus Fusarium xyrophyllum create on xyrus plants? Pseudoflower. Woohoo. Yep. And there are other fungi that make pseudoflowers, some that even make nectar, which is on Heath here in Washington, on Erica. Yeah, pretty crazy. I had a hard time thinking of more questions. Lichens in the tropics, I really like to grow on. Then there was a certain substrate I mentioned. I don't know if I put an emphasis on it. Palms, woohoo, you got it. Yay. Palm leaves. There's tons of palms in the tropics. If you didn't guess that, it's not a stereotype, but that certainly stands true. Lots and lots of palms. Some that have crazy spikes all over every part of them, and they'll get you if you don't watch out. All right, that's that. Happy to take any questions. Let me stop sharing and see what happens here. Okay, now we have this cool meta thing going on. Okay, great. Here you go. I'm gonna peek at this. Thank you all for being here. I really loved sharing this with you.
We do have a question in the audience.
First of all, I thought that was a terrific presentation. As someone who's not really engaged in biology at a research level, I was wondering, you discussed that the pseudoflowers are still kind of trying to identify their role and the ecology of that. And could you sort of maybe just give a high level overview of the scientific process they use to sort of suss that out and figure out what's going on there?
So the main things we were collecting and figuring it out, we were collecting soil samples, trying to see, is the fungus present loosely in the soil, plant material, trying to also see, we're trying to find out where is the fungus living. Besides, we know it's making the pseudo flower, but where else is it? Because that's going to help us look at its life cycle and see how it's spread. So collecting soil, and Terry has experience, that's part of her PhD, is looking at biogeochemistry. So she's analyzing the soil. I think she's also trying to figure out like what the soil is composed of, trying to see like if there's any correlation there with where the plants are growing. And she's looking at the Xyrus and the Fusarium interacting, but there's also not a lot known about Xyrus in general and Xyrus insect interactions as well. So she's also looking at kind of what insects are present, trying to identify them, seeing how they're, are they present on the pseudoflower? Are they present on the flower? And then trying to identify those, categorize those. And then she's also was collecting tissue for microbiome samples. So she's going to get all this genetic data back, trying to tell us, are there yeasts, are there bacteria, are there other fungi that are living within the plants and the pseudoflower that maybe have a role that we can't see macroscopically. And then she was collecting volatiles. So we were creating like a little suctioned airspace around the plants, and then we'd make these tiny bouquets, and to have kind of more concentrated data, where she had a pump pulling the air through, and then that's going to go, and then we get a little vial, and that's going to go to a chemistry lab, and she's going to work with people to figure out what compounds are being emitted into the air that are produced by the fungus, produced by the Xyrus, and then are there overlap? Is that part of what's luring insects in? So it's a pretty like kind of getting data anywhere she can to try to create a story and a picture of what's going on there. So yeah, I think that's mostly what we were collecting: tissue for microbiome, volatiles, soil, insects. Yeah, does that better answer? Yeah, of course. Oh yeah, let's see. Maria, hi Maria. Did you get a sense about why that curator at the herbarium refused samples of fungi? That's a good question. It seemed like really political and very personal for this person. I think it was somebody who got the position through nepotism and wasn't really qualified for it. And then just had the idea that they were going to run the herbarium a certain way. And, fungi aren't plants they're only more recently known to not be plants, but I think she probably saw it as being too irrelevant to the, what they were doing at the herbarium. But there was the space and the time for people to like document these fungi there, like they had the capacity for it, but she was just wanted to maintain a certain image and kind of assert power over the situation, is how it seemed, unfortunately. And some like this one person who came in and was really passionate about it kind of didn't have another place to go or another person to talk to. But she reached out to mycologists and learned a lot in her time. And she's a really skilled botanist who still loves mushrooms. So it didn't kill her love for fungi. And she's working on getting her specimens put back into the herbarium. Yeah. Any questions more in person? Hello, Ben.
Did you encounter?
Oh, sorry.
I recall you had mentioned there was a mycoheterotrophic plant that you had seen, and I was just curious. First of all, was it Xyrus? Sorry, that was the name of the genus of the plant that you said it was not in grass. It wasn't a grass, but was it what plant family was it in? And sorry.
No. We're getting used to this all. That's a good question. Ben was just asking where does the Xyrus belong taxonomically? So it has its own family, Xyridaceae, and it's in Poales. Poales is the grass order. So I was talking about it, and a really experienced biologist was like, it's not a grass. So now I'm like, it's not a grass, but it makes these spikes and it doesn't make like a grass-like inflorescence. So it's kind of its own family. And I don't really know somebody told me it was more kind of closely related to irises, but I looked into that and that doesn't seem to be the case. So I don't exactly know what the next closest relative is, but they're believed to be, the tropics is thought to maybe be the center of diversity for its iris. So
Kind of like a second part of that, which was,
sure. I can just reiterate, yeah, second part of the question.
did you find this, any other types of, well, obviously that wasn't a mycoheterotrope, but I was kind of in my head wondering about these plant and fungi relationships. Like, was there a specific plant family that seemed to have, how we have a lot of ericaceous
Yeah.
mycoheterotropes?
Yeah, I'll repeat this so shortly.
I was curious, if there was some kind of trends that you were seeing, regarding those.
Good question. Pen's asking if there was any other trends like in the Pacific Northwest, and we have a lot of ericaceous plants that have microheterotrophic relationships. So that's when the fungus derives its nutrients from a fungus that's connected to plants that's getting its sugar that way. So it kind of hijacks. And for this tropic strip, we really, as far as I know, I didn't recognize even any other besides in Trinidad seeing, I think, 1 or 2 plants that were mycoheterotrophs. I don't think I saw any other like direct integrated relationships between the plant and fungi like that. But that's also only like to what I know. And I had never been to, on a research trip to these places. So I might have seen this right in front of my eyes. And like with the pseudoflowers, there were botanists studying Xyris who collected the pseudoflower thinking it was a weird Xyris. And they had been studying this plant. So for all we know, I could have been, and they fooled people and they're like, this one looks weird, so I could have been collecting something, and that's how science goes. So maybe someone comes along and says, look, maybe I collected something that people will find out was a weird interaction between plants or 2 fungi or something. But unfortunately, I don't have another example. Yeah, thank you. Any other questions? Let me see if there's any more here. Somebody asked about the foray. Is it possible to just come out for the day and join the spring foray, or do we have to stay at the lodge? Oh, for the morel foray. Yeah, I don't know if we have worked that out yet, but if you contact, respond to the emails and the stuff, and we'll see what we can do to work with you. Okay, and then I got a private message. When's your book coming out, Lauren? Beautiful pics, great presentation. Thanks, Bo. And then Kirsten says, how did you manage to go to Trinidad on a mushroom collecting trip? This was a super interesting plus mind-blowing talk, Lauren. Thank you, Kirsten. How did I manage to go to Trinidad? So Trinidad is a common layover if you're flying to Guyana. It's pretty close to Guyana. Like I said, Guyana borders Venezuela, Trinidad's off the coast of Venezuela, and there's an airport that you frequently stop at. And so we were looking to buy our flights, and Jack's like, doesn't Luca have family in Trinidad? What if we go to Trinidad? What if we try to make that happen? And we start chatting with Luca, and their mom wanted to go down there anyways to see family. And so we made a trip happen, and we stayed in a full packed family with 3 kids and 4 of us staying there, and the 2 parents in a little townhome. And it was great. And they have a lot of community relations, and it's like going to Trinidad, seeing how much people know each other and look out for each other was really cool. So we were able to go to the Savannah, work with the college. That was done partially on our own, contacting the herbarium people, but we just kind of got, part of it was working to make it happen, and part of it was getting lucky with the connections that people had. Getting to go to these beautiful sites, some beautiful waterfalls and hikes and stuff, Coco Palace. But I recommend going. It's actually pretty affordable to go to Trinidad. If you're going to go somewhere tropical, cool, go to Trinidad. People are really awesome, and there's good food. Okay, any other questions? Hopefully that answered that. It was just kind of, it feels like it was a whirlwind thing that ended up coming together. I'm amazed that it fit the way it did. But yeah, I've been talking a lot. I guess that's why I'm here. Okay, guys. Oh, yeah, we were gonna do raffle, and there's one more thing that popped up in the chat. Oh, yes, you did. Did you eat some mushrooms? Nope. I didn't eat a single mushroom. I don't think. I actually don't eat that many mushrooms. I like to eat them, but I'm a little bit picky. We did find one agaricus that smelled like almonds, just like the prince does, but we had no idea if it was edible, so we didn't eat it. It was in great condition though, and really beautiful. Yeah, I don't think I ate any. I ate a lot of French fries. And both Guyana and Trinidad have a really, really large Indian population. And a lot of people don't know that. And so it's this like Afro-Caribbean, West Indian cultural mix. And it's like unbelievable, like to see how that affects the language and the culture and the food. And there's a lot of really incredible Indian food there. If you go, get doubles. They're really good. They're like tacos, but they're Indian. Okay. And yeah, okay, raffle. We're doing the raffle.
All right.
These are pretty tickets.
So everyone who showed up in person might win a prize. We have a lot of prizes, so go ahead.
Woohoo, I get to do- You guys got your numbers out? Well done. They're stuck together. Okay. 03.
Just go 903.
Oh, 903.
Woo! Okay. That table over there?
How many are we doing?
I don't know. Let's keep going. Let's do a bunch.
906.
Woo!
You already got a prize.
Winner, winner.
Great. Yeah, go get your, we believe you. Okay, here we go. 908. We're pretty consecutive. Oh no, we're not. 908. Yay.
Woo! Let's do two more.
Okay.
And maybe we'll do even more. I don't know.
915. Wait, who has it? We got two. Now we might have
Are there
to check.
two 915s?
Wait. Oh no.
Oh, never mind.
Okay.
Okay, one more.
Oops. That's the one.
Yeah, that'll be it.
901.
Woo! Okay, should we keep going? We got a lot more prize. Let's keep going.
Yeah, you guys.
You know what?
rock for
We
showing up.
really want people to start coming in person, and we're going to be doing this every month. Let's do 5 more. Let's give away 10 raffle prizes. It's pretty cool. We have a really cool microscope. We've got a plate. We've got some original artwork. We got some lapel pins. We've got the club lapel pins too. Some little Christmas ornaments. 902.
902.
Melody won.
What are we at right now? We're at 1, 2, 3, 4, 5, 6.
Here, we'll do that one.
Yeah.
918. Justin. 914. Woo! How many do we do? I forgot how many.
2, 3, 4, 5, 6, 7, 8.
Okay, so we're going to do 2 more prizes. The next one is 917. Oops, and then the one that fell on the floor, 905. And this one fell on the floor too. Let's do 11. Why not? 913. So we got 905 and 913.
Oh, okay. So skip that one.
Oh, did it go back in? How do we have
I don't
2?
know how that happened.
Oh, it's 905. I'm sorry. It was 905. Whatever. You can both get a prize. Go pick one. We're just giving away prizes like crazy today. Okay, cool. All right. Congratulations to the winners.
See you Friday if you can come to the foray. We will poke around at tiny things and have a great time.
And for those of you who came in late and missed the intro, we've got, is it April 5th, Elio Schechter? Dr. Elia Schechter for NAMA. If you're a member of NAMA, the 6th. Hold on. I don't want to give you the wrong date.
It's the 6th.
Okay, April 6th. And that was a lecture I helped put together, and I got to meet him. He lives in San Diego, and he founded the San Diego Mycological Society in the 90s. And he's absolutely amazing, and he's super funny. And it's going to be a great lecture, and you guys should all watch it. It's going to be on Zoom. Then this Friday, we have a foray at Ptolemy State Park. It's just going to be an ecology walk. We're not going to find, well, maybe we will, but I doubt we'll find any good edibles. But it'll be really fun. And then we've got April 30th, the Spring Mushroom Festival. It's going to be right here. And we'll have more info shortly. Sorry, I know it's short notice. We don't have everything totally set in stone yet. And then was it May? Save the date. It's the last Saturday in April. It's the last day in April. And then May 20th, 21st. Yeah, May 20th, 21st, and 22nd, we have our big morel foray. It's going to be near Cluellum, and we are staying in a really nice place with beds and showers and 3 meals a day. So it's not going to be camping this year. Although I do believe is RV camping an option. You can camp if you want. And check your emails that was mailed to all of our members. There's a survey too. Yes. If you just joined and you didn't get the email, just email the website and ask for it. Or if you email the social media, I can forward it to you. If you email the website, Steve Ferguson can email it to you. So one of the volunteers can do it. We're at South Sound Mushrooms on Instagram, Facebook, and Reddit. So follow us on social media.
Cool.
All right, that's it, I think.
Yeah. And we'll send out more reminders for everything too. Yeah, thanks guys. Have a great night. And we'll see you soon in some way or another. Look for mushrooms always. Put them on iNet.